Good morning to all of you. So, today we will start with the next topic of the first chapter of biotechnology, principles and processes. So, the topic which we finished yesterday, it was about the first recombinant DNA, which was formed by, first of all, synthesized by Stanley Cohen and Boyer, Herbert Boyer in the year 1972, and the two microorganisms which were used in that, those were Salmonella typhimurium and E. coli. So they used it, they made it, and this was done in the year 1972. Then yesterday we did how it is possible to multiply the desired gene in the host, as well as how to get it incorporated in the progeny. Means how will it come in the progeny? Then only we can say that fine, this recombination was successful. So this we did yesterday. Today we are going to start with the enzymes. Enzymes which are required. Today's topic is enzymes which are required, which can be considered as a tool of recombinant DNA technology. So we are basically starting with the topic tools of recombinant DNA technology. This is the topic tools of recombinant DNA technology, and today the subtopic which we will take up is enzymes. You know, various enzymes are very important in the process of biotechnology. Various enzymes, and we will take up all the enzymes which are required in the process of recombinant DNA technology, and I'll tell you these enzymes one by one. The first enzyme which is required in forming a recombinant DNA or in the process of recombinant DNA technology are the lysing enzymes. The first type of enzymes are lysing enzymes, L-Y-S-I-N-G, lysing enzymes. This word lysing has come from a word which we have discussed many a times in class 11, and the word was lysis. Anybody who remembers the meaning of lysis? Yes. Anybody who remembers the meaning of lysis? On that basis only this enzyme lysine has come. Yes, what is the meaning of lysis? Breakdown. Yes, Suksaha, Jamit Katoch, all are answering correctly. It's breakdown. So breakdown. Now why this breakdown is required in recombinant DNA technology? This breakdown is required because we have to obtain the desired gene. For making a rDNA or recombinant DNA, we need the desired gene. And from where we will obtain the desired gene? The desired gene from where we will obtain it? From where it is obtained? It can be obtained from a cell. Now, it's not that it is kept at the periphery of the cell or on the membrane or on the cell wall of the cell. It's not that. We have to obtain it from a deep-seated organelle, which is nucleus. So this desired gene has to be obtained from the nuclear material. This desired gene has to be obtained from the nuclear material. You know, the DNA is in the nuclear material, and we will obtain this nuclear material from the nucleus. So the desired gene which we want to with which we want to make rDNA or recombinant DNA has to be obtained. And from where we will obtain it? we will obtain it from the nucleus, and specifically the nuclear material. So how can we break the cell? For that we will have to break the cell, obviously. So for breaking the cell, some enzymes are required, which we call as lysing enzyme. Lysing enzyme, or the enzymes which bring about lysis of the cell. So what will be done here? The cell from which we will obtain the genetic material or the desired gene. It can be an animal cell, it can be an animal cell if we are doing it for the animals. It can be, we can get it from an animal cell, that desired gene. It can be a plant cell, it can be a fungal cell of a fungus from where we can obtain the gene, or it can be a prokaryotic cell. That means bacteria. Here, by this I mean bacteria. So the desired gene can be obtained from an animal cell, the plant cell, a fungal cell, a prokaryotic cell. If you remember, when we were doing the second chapter of biotechnology, when we were doing the use of biotechnology in medicine, we did how hirudin is prepared. And for that, we obtained the desired gene from the leech, and then we introduced it or inserted in a plant, which was Brassica napus, if you remember. I think you all remember this. So the desired gene was taken from one animal and was introduced into a plant. So sometimes we use an animal cell, sometimes a plant cell, sometimes a fungal cell, or sometimes a bacterial or a prokaryotic cell. And in different cases, we have to use different lysing enzyme, because all these cells, they have different type of envelopes, they have different type of cell envelopes, different type of covering around them. If we come to the first type, which is animal cell, in the case of animal cell, there is just one outer covering, and that outer covering is cell membrane or plasma membrane. So in the first case, we have the plasma membrane, and this plasma membrane, what are the components of plasma membrane, if anybody remembers? What are the components of plasma membrane, beta? We have done this in 11th class, yes. What are the components of plasma membrane? Anybody? The only envelope which is present around the cell, which has to be broken to get the genetic material, is plasma membrane. Now, what is a plasma membrane made up of? If you remember, we did fluid mosaic model, lipids and proteins. That's right, Sukhsahaj, lipids and proteins. So we need some enzymes which breaks up this lipids and protein, because plasma membrane is made up of lipids and proteins. This you all know. So we need some enzymes which breaks down the lipids and which breaks down the proteins, and we'll be able to bring about the lysis of plasma membrane. So which enzymes do we need as lysing enzymes? Here in this case, we need proteases and we need lipases. So these two enzymes are required, which are proteases and lipases, to break down the plasma membrane, which is the only envelope present in the animal cell. And we'll be able to bring about the lysis of that cell to obtain the desired gene. Then the second one. Second is the plant cell. Plant cell has two envelopes. Plant cell has two covers. The outer one is cell wall, and then the inner one is cell membrane. If you remember, the cell wall is made up of, beta? Yes, cell wall is made up of, cell wall in plant cells is made up of which material? Yes, yes, anybody answer? Cellulose. So Nidhi is answering cellulose. That's right. So yes, Suksa had cellulose. Yes. So we want another enzyme to break that wall which is made, which is the cell wall which is present in the plant cell. So we need the enzyme cellulose. Cellulose is broken down by cellulase. Cellulase. This enzyme is required, and besides this enzyme, in some cases, what happens that the besides cellulose, pectin is also present. If you remember where the secondary growth takes place, pectin is also present, which is a hard substance. So, besides cellulose, pectinase is also required. Pectinase. So now the cell wall of the plant cell is broken. And inside the cell wall, what do we have in plant cell? The same cell membrane, which is made up of lipids and proteins. So now we also need proteases. We also need lipases. So these are also required to bring about the lysis of the plant cell. Because in plant cells, we have two covers, the outer cell wall and the inner cell membrane made up of proteins and lipids. Then comes the fungal cell. If we want to obtain the material from fungal cell. So fungal cell, see, fungus was first kept in the plant kingdom. Why was during the classification fungi was a group of plant kingdom. It was included in the plant kingdom because of the presence of cell wall. Later on, because it was not autotrophic in nature, it could not prepare its own food, it was separated by Whitaker. If you remember, Whitaker then separated it, made a separate group, a separate kingdom fungi. But before it, it was included in the plant kingdom because of presence of cell wall. Then later on, Whitaker said, no, it is not autotrophic in nature. It cannot synthesize its own food. So it should be kept in another group. And he excluded it from plant kingdom and kept it in kingdom fungi. So fungal cell, like plant cell, also has two coverings. The outer one is wall and the inner one is membrane. So in case of fungi, the material of cell wall, what is it? In case of plant cells, it's cellulose. But cell wall in fungi, in fungal cell, is made up of, yes, cell wall of fungus. Anybody remembers? Cell wall of a fungal cell. Yes, the chemical nature of cell wall of fungi. Yes, anybody remember? Anybody remembers? It is chitin. It is chitin. Yes, Suksahaj, yes, it is chitin. So, what do we need to break that cell wall? We need chitinase. We need chitinase. So, another enzyme which is used here is chitinase. And along with chitinase, inside again proteases and lipases. We have this. Now we come to prokaryotic cell. If the desired gene has to be obtained from a prokaryotic cell, then what do we need? We need an enzyme which is known as lysozyme. So only lysozyme can do the needful of breaking up of the wall of a prokaryotic cell and helping us to obtain that enzyme. Now, obtain the genetic material. Now, these were the first set of enzyme which was required in the process of recombinant DNA technology. Now, today we are doing all the enzymes which are required. So, the second enzyme which is required is alkaline phosphatase. This is, of course, this alkaline phosphatase is not that important. Restriction endonuclease or endonucleases are more important, but that I'll have to explain in detail. So I'll take endonucleases at last, because endonucleases has to be explained in detail. So I'll take endonucleases at last, which is also an enzyme which is required for recombinant DNA technology. I'll take that at last. Right now, I'll take alkaline phosphatase, which is an enzyme required. Alkaline phosphatase. This is another enzyme which is required for the process of recombinant DNA technology. And what is its function? Suppose you have taken a plasmid here. This plasmid you have taken to make a recombinant DNA. This is a plasmid you have taken. Now, what you do to insert the desired gene into this plasmid? How the desired gene is inserted into this plasmid? For inserting the desired gene in this plasmid, you cut it. And that cut is made by molecular scissors. I'm not taking the name right now, endonucleases, because I have not explained endonucleases to you. I'm just using molecular scissors. So molecular scissors, these are required to make a cut. Now, when a cut is made, it will come in linear shape. It will take a linear shape, and because it is a genetic material DNA, it will have 3' and 5'. Okay, 3' end has OH and 5' has phosphate. 3' end has hydroxyl group and 5' has phosphate. This is what we will obtain. Now what happens? You know, this is the nature of hydroxyl group and phosphate to join and form phosphoester or phosphodiester bond again. So these two, OH and phosphorus, they join to form phospho, you can say diester or phosphoester bond. This bond is formed. Now what happens? If once it is cut, it comes into the linear shape, but it tends to roll again. It tends to roll again because this is, you know, the tendency of hydroxyl group and phosphate to form phosphodiester bond. So for a while, it will come in the linear shape, and again it will roll and form. It will come in this shape. Again, it will come in this shape, and our task will not be successful. So what is done? So that it does not form phosphodiester bond again, it does not roll back and form phosphodiester bond again, this alkaline phosphatase is used. This alkaline phosphatase is used, and this enzyme, alkaline phosphatase, what it does, it removes this phosphate. This phosphate is removed. It removes the phosphate. Once the phosphate is removed, it cannot form phosphodiester bond, so it will remain in the linear position. It will remain in the linear shape. So now what do we get? We get this 3' end having OH, 5' end without phosphate, no phosphate. Okay, no phosphate, so that it does not roll back. It does not roll back and form phosphodiester bond, because what happens immediately after, it will, this straight one, it will roll back and form the phosphodiester bond. So it's of no use, you know, using molecular scissors then, because it will roll back and form phosphodiester bond. So alkaline phosphatase is used where phosphate is removed, so that it does not form a phosphodiester bond with hydroxyl group. Now, to this end, the desired gene is attached. So, the desired gene is attached to this end. Like, suppose we have this desired gene. This desired gene is attached here. And after attachment, you know, again this phosphate is added. How this phosphate is added, that we'll do it in the next enzyme. Okay, so this is how the desired gene is then added in the linear position. But you know, something which is kept in a linear shape inside an organism is better, or something which is in a circular shape inside an organism is better? What do you think? Which one is better? Which one is better? Which will cause less problem? Which type of recombinant DNA will be better? In linear shape or in a circular shape? Yes. What do you think? Circular. Yes. Paras Singh is saying. Sunidhi is also saying circular. Yes. Yes, circular is better. So that is why later on again this removed phosphate is added and it is brought back to circular shape and then introduced through vector. Okay. So this is what is done by alkaline phosphatase, that the phosphate is removed so that it remains in the linear position and the desired gene is easily attached to it. Later on, of course, it again comes back to circular position. How? This I'll let you know when we will study the next enzyme. So now this was the second enzyme, which is alkaline phosphatase. We come to the next enzyme. Okay, now one more enzyme is very important. Today we are doing enzymes, so I'll tell you all the enzymes which are used in DNA technology. So the next enzyme, which is very important, the next enzyme which is very important is ligase, the third enzyme. It is enzyme ligase. Now, what's the use of this? What's the use of this DNA desired gene if it will not attach to it? If it will not attach to the this particular plasmid, what's the use? It should attach properly so that a proper recombinant DNA is formed, rDNA is formed. So we have done this enzyme ligase before also at the time of when we studied replication of DNA. All that we have done. Okazaki fragments. At that time, we have done the use of ligase, enzyme ligase. So here also, see this red colored is the desired gene, and this is the linear shaped plasmid which is made. But what's the use if this doesn't get attached to it properly? It should attach to the plasmid properly. And this attachment is brought about by enzyme ligase. So this is the enzyme which attaches or joins the desired gene to the plasmid and which results in the formation of rDNA. This is the enzyme which is required and which is of great importance so that the desired gene is attached to the plasmid. Now this was next enzyme, third enzyme. The fourth enzyme is phosphorylase. The fourth important enzyme as a tool of recombinant DNA technology is phosphorylase. Phosphorylase, or sometimes people call it phosphorylase also. Now, what is the function of this? This is opposite or antagonistic in function to alkaline phosphatase. Alkaline phosphatase removed the phosphate group. Now this phosphorylase, this will help in addition of phosphate. This phosphate was removed. If you remember here, it was removed and we had OH here, and at 5 prime we did not have phosphate. So what happened that it could not roll back. So now this phosphorylase, it helps in the addition of phosphate. This helps in the addition of phosphate. Now what is the importance? Why was it first removed and then added? Why was it removed? That I have already told you, because it was easy. What would have happened that the plasmid we wanted in a linear position so that the desired gene can be introduced, inserted and attached And now it is rather difficult, it is rather not advised to put this recombinant DNA in this form inside the organism, but the recombinant DNA should be in a circular form. So for the formation of circular form, for circular form, so that it has to be, so that it can be brought in circular form, there should be formation of phosphodiester bond. And when this phosphodiester bond will be made, it will be made when there is hydroxyl as well as phosphate at both the at one end hydroxyl and another end phosphate. So it will be possible in that case. So for that, the phosphate has to be added, and this enzyme phosphorylase helps in addition of phosphate at 5 prime. Now we have OH at 3 prime and 5 prime we have phosphate. Now what will happen? Both of this will roll back and both OH and P will form phosphodiester or phosphoester bond, and ultimately we will get it back in circular form like this, recombinant DNA with the gene here, with the gene here. Is it clear, these 4 enzymes which I have told you just now? Is it clear, the functions, the importance of these 4 enzymes in DNA technology? Is it clear? Any question or any portion you want me to repeat, or it is clear, beta? Yes? Yes, I want your response. Is it clear? These all 4 enzymes? Yes, bache? I'm not getting any response. I'm not getting any response. Yes, ma'am. What is the term? Antagonistic, beta. Antagonistic. The term used is antagonistic. Antagonistic in action. When we did the endocrine glands, at that time when we were doing insulin and glucagon, that time also we use the term antagonistic in action. Antagonistic. Okay. Any other thing from this, or I shall start with endonucleases? Okay. Yes. So I think this is clear to you, these 4 enzymes. We start with the next enzyme, which is very important enzyme. Very, very important. Without that, the recombinant DNA technology is not possible. And that enzyme is endonuclease. So beta, we start with the next endonuclease. So this is the 5th enzyme, which is endonuclease. This was first of all, found out in the year 1963. These endonucleases, when the researchers found that this enzyme endonuclease, it has 2 parts. It has 2 parts. One was methylated agent, and another part was endonuclease. Methylated agent, and the second part is endonuclease. Now, what is this methylated agent? How it helps in the endonuclease? First of all, what is endonuclease? Endonucleases are also known as molecular or biological scissors. Molecular or biological scissors. When we say scissors, of course, they have the function of cutting at a specific site. Now we have to be very clear. They cut the DNA at specific site. They cut the DNA at specific site. That is why they are known as restriction endonucleases. They are restricted to make a cut on DNA at a specific site. What is that specific site? Suppose there is a particular endonuclease or restriction endonuclease which can cut between C and G. Here it can make a cut here. Then we can have some endonuclease which can cut between A and T. Then we can have which can cut between G and C and all this way. So we have these endonucleases which cuts, which makes a cut at DNA on DNA at specific site. Now, how these two complexes, methylated agent and endonucleases work. Now, suppose we have, let's take an example of a prokaryotic cell. We have this prokaryotic cell, say a bacterial cell or E. coli, because E. coli is used a lot in the study of biotechnology. Let's say that we have E. coli here. Now, this E. coli is infected with a bacteriophage. What is a bacteriophage, beta? This is infected by a bacteriophage. Yes, bacteriophage, what is it? What is bacteriophage? Yes, anybody? What is bacteriophage? Virus. Yes, Paras Singh is saying virus. Specifically, this is the virus which attacks bacteria. So there is a virus, a bacteriophage, which is attacking this virus, attacking the bacteria. So this is a bacteriophage which is attacking the bacteria. Now what is inserted from the body of the bacteriophage? It is the genetic material which is inserted. Now, this bacteria has its own DNA. It has its own DNA. Suppose the circular DNA belongs to bacteria. Okay. Now, from the bacteriophage, its genetic material is injected into this. Now, we are using a endonuclease which cuts here. Suppose we are using a endonuclease which cuts in between this side, that is between cytosine and guanine. And this is how this defensive mechanism is going to take place. That means we have the endonuclease, restriction endonuclease, which has this methylated part, methylated agent, and we has endonuclease part, both. So I told you this endonuclease, it can cut at C and G, in between C and G. This is how it is going to deal with the genetic material of a bacteriophage. And suppose it has C here and it has G here. The same C and G, cytosine and guanine, is also present in this. I'll make it with another color so that you are able to see it. Suppose the same, because nitrogenous bases are same. So the nitrogenous base is present here also. Here also. That means the nitrogenous base is